Today we will be discussing more on polymerase chain reaction. We had some introductory discussion on the polymerase chain reaction. Today we'll take the discussion a little bit further and we'll discuss something more about polymerase chain reactions, such as the general layout of the reaction and so on. We'll start with the amplification. Amplification means the DNA molecule is being amplified, but what is the factor that is responsible? In this case, we have DNA polymerase. And DNA polymerase, as you can see, the name contains three important components: DNA, poly, and merase. Poly means many, and then merase, and in that one, A's word is for enzyme. So it's the enzyme that can make many DNA molecule. In simple word, if you want to recall or remember this. So and what is DNA polymerase? It's nothing but the protein molecule, and protein molecules, we all know that they have the three-dimensional structure, which is important for their function. Now, DNA polymerase is also known as Taq DNA polymerase. Why? Because it is extracted from, or it is purified from, Thermus aquaticus. That is the name of the organism, which in short we can call it as a Taq, right? T, Thermus, AQ is the coming from the aquaticus. Now, this is the microorganism that is surviving under the very, very hot environmental condition in the hot springs. It can survive the high temperature, and that is very, very useful for the polymerase chain reaction, because we have the denaturation step at the start of the PCR, where we want to separate the DNA molecules. We want to basically separate the DNA strand of one DNA molecule and then perform the further steps in PCR. Next is, it is suitable for PCR because you have the high temperature, which is 94 degrees centigrade, and these proteins, they can survive those high temperatures during the polymerase chain reaction. The first step is denaturation. Now, Taq DNA polymerase is resistant to denaturation. It is resistant to denaturation. And what is denaturation? What happens is that 3D structure of the protein, if you expose that structure to high temperature or heat, what will happen? The molecules, they're going to get fragmented, and we can call that the molecule has been degraded. So the process of degradation caused the molecule to become fragmented, and now it's non-functional. So it will no longer be functional. But Taq DNA polymerase is resistant to heat. Now let's move on to the general layout of the PCR reaction, where you have four important components. I'll just broadly classify them in different parts. One, DNA. Second one, so you have the template DNA. In this case, second one is the primers. Third one, DNA pol, which is DNA polymerase. Fourth one is the additional factors. In additional factors, you have buffers, you have DNTPs, because you want to amplify the molecule. So we'll start from the DNA. DNA is nothing but the double-stranded DNA molecule in this case, and a very, very small quantity is good enough, and one DNA is basically good enough for the polymerase chain reaction. You might have to increase the cycles, but yes, if you amplify and then you can repeat your cycles, one DNA is quite good enough. Next is primers. Primers are short DNA molecules, and they have the tendency to bind to the complementary sequences, and without that, DNA polymerase cannot function because they require these small oligonucleotides. Next is additional factors. You need buffers for the proper functioning of the DNA polymerase. You need DNTPs so that DNA polymerase can add DNTPs and the DNA can be constructed. Next is, if you see, you will have a PCR tube, and I'm trying to design the PCR tube here. So something like this is the general layout of the PCR tube, where it is a little bit smaller than the traditional Eppen drop or the centrifuge tube, which is smaller one, 2 ml Eppen drop tube, where you will add all the components. The reaction mixture is ready. First step is to heat the reaction mixture to denature the DNA, where the hydrogen bonds between two strands, they will be broken down, and then the bonds, once the bonds they are broken down, what will happen? The strand will be separated out. Now, joining of the primer is the next step, where the temperature will be decreased to 50 to 60 degrees Celsius, and after that, what will happen is you can increase the temperature again so that DNA polymerase can function. So the DNA polymerase will function at 72 degrees centigrade. So that's the optimum temperature. So this is, I think, this covers most of the important points regarding the the polymerase chain reaction. And we'll continue our discussion on that and explain these things in a little bit more detail. So please watch the next video. So let's summarize what we have discussed in this one. We discussed that how the amplification happens, what is DNA polymerase, how it functions, and then why we are using Taq DNA polymerase, what are the different components of polymerase chain reaction, and why they are important. So I hope that you got some general idea about the polymerase chain reaction. If you want to study more, you can read books and you can see online. There are a lot of information. There is a lot of information that is available. There are a lot of articles available that you can read, and you will find a lot of good information on polymerase chain reaction. All right, I'll meet you in the next video where I will again try to explain a little bit more details of the polymerase chain reaction. Till then, take care.